neurotransmitters and receptors. So I will go ahead and share my screen. Okay, so here's your outline, which you have, which is already posted on D2L. So we'll talk about receptor systems first, major neurotransmitter systems, and next lecture will be how this relates to drugs, both psychiatric and drugs of abuse. So you have two main receptor systems in the central nervous system. You have ionotropic receptors and metabotropic receptors. The first one I'm showing you is ionotropic receptors. These have binding sites, or these have receptors, which are directly tied to the ion channel. Remember, I said if you learn nothing in this course, know that neurotransmitters don't go into the cell. We know from last lecture with the action potential that ion channels open and it changes the membrane potential of the cell. So essentially, when a molecule here, the circle comes and binds, it directly opens the channel. That's what you're seeing down here as well, that the channel opens. So it makes it quick communication because there's no middle mechanism, and it usually doesn't last that long. It's pretty quick. So here is exact sort of, and it's a lock and key model, remember. So specific neurotransmitter goes to a specific receptor, right? So it's just something like that, and then it will open the ion channel. So the receptor itself is on the ion channel. And in the neuroscience, there's multiple systems. This is called ligand gated as well. So neurotransmitter would come, it would sit here, and if it opens, let's say here it's opening our sodium channel ever so slightly. If sodium flows in, think about whether or not that's going to lead to an EPSP or an IPSP. So excitatory postsynaptic potential or inhibitory postsynaptic potential. So it makes the inside of the cell more positive, then it's going to move us towards an action potential, towards that positive 40, or towards, I guess, first that threshold of negative 50, and then move us up towards positive 40 if an action potential occurs. So if this moves us closer to an action potential, then it's going to be an EPSP. Whereas chloride here, if we open this and it goes into the cell, it will make it more negative. And if it makes it more negative, it moves us farther away from the action potential, and then that's an IPSP. So if this is negative 70, where the resting potential is, an EPSP will move you up, and an IPSP will move you down and farther away from an action potential. So they can be both excitatory and inhibitory. Now you notice on metabotropic receptors here, we already have some other things. And note that the ion channel there in brown is not close to the receptor there in blue. And as I mentioned, there's multiple names for everything. Sometimes this is called second messenger systems, because you will see that there's a second messenger. And sometimes it's called G protein coupled receptors, which mainly, if you choose articles, they'll likely call G protein coupled receptors. So you're going to have your neurotransmitter. It's going to bind in our lock and key model, but you have a trimer of G proteins, which means you have 3 G proteins at the bottom. One will dissociate, usually the alpha subunit, go over to the ion channel, and then it opens. So because of this, these are usually slower acting than ionotropic receptors, and they usually last longer because you have a change in those intracellular processes that occur. So one more time, we'll do it. Triangle neurotransmitter goes to triangle receptor. You have the alpha unit, which dissociates, and it opens the ion channel, which is somewhere else on the cell. It's not directly tied. If it's directly tied, ionotropic. If it's somewhere else in a G protein, trimer of G proteins are involved, then it is a metabotropic or a G protein coupled receptor. So here is just from the book. You have here your metabotropic receptor. So again, the receptor, a G protein, the alpha subunit breaks away and then opens the ion channel, which is somewhere else, right? There is another kind of metabotropic receptor. I just want you to know this exists, but I'm not going to ask you about any kind of detail. That here you have a G protein, it comes away, it can activate an enzyme, which activates a second messenger system, and then that's what's going to go and open the ion channel. And it also goes into the cell and creates a cascade of long-term effects. So because of that, metabotropic receptors, because they have some of these intracellular process changes and second messenger, that they're usually longer acting, their effects. And a lot of our like catecholamines and things like that are going to use a second messenger system. So if neurotransmitters comes and binds to those receptors and opens the ion channel, then we can either have an EPSP or an IPSP. And again, because I told you that neurotransmitters don't get sucked up by the cell, they just keep binding over and over and over again. So it keeps being an EPSP or an IPSP. So how does that happen? Like how can you stop there being an EPSP or an IPSP? How can you stop there being a postsynaptic potential change? How can you just keep that neuron at rest? Well, there's 3 ways. One, which is not written on here, but the first one is diffusion. So I'll actually write that. Okay, are stopped by 1 diffusion, which means they just go away. We already know about a diffusion gradient. Things want to move for where they're really concentrated to their less. So they just move away and get taken out maybe by phagocytosis or something somewhere else. They could also be reuptaken into the presynaptic cells. This is the presynaptic cell here, which would make this the axon terminal, which would make this the dendrite. So it can be taken up by a transporter. So that is a transporter that is not an autoreceptor. An autoreceptor is a receptor which will bind here and give feedback to the cell about how much of it is in the synapse. This actually takes it up into the synapse. Or it can get broken down by enzymes. So here you have an enzyme which lives in the synapse called acetylcholine esterase. It breaks acetylcholine down into its 2 parts, acetate and choline, and then it can't bind again, and so it's inactivated. And that usually will get retaken up by the cell or go away by diffusion. So those are the 3 ways in which you stop a postsynaptic potential from happening, either an EPSP or an IPSP, because a neurotransmitter will bind over and over again unless something moves it out of the synapse, essentially. So, what do I want to say about this? So we're going to talk about how things sort of activate receptor systems. And we're going to use the terms agonist or antagonist for how they activate certain systems. So let me talk about it, and then you have an in-class assignment that is posted for you. So if you have something like an agonist, it's something that facilitates the activity of that neurotransmitter system. So that means it has the neurotransmitter system do what it would normally do, right? So if dopamine binds on a dopaminergic receptor and it opens an ion channel, that's an agonist to that system because it's doing what it would always do. And it doesn't have to happen at the level of receptor. It can happen earlier. So for example, right here, maybe you have these neurotransmitters here in synaptic vesicles. We know that that calcium, those voltage gain channels open, calcium comes in and it fuses the vesicle with this membrane here. That is how neurotransmitters leave the cell. There are drugs that will get rid and degrade these vesicles. If that happens, then it can't leave the cell and it can't come down here to affect the system. If it does that, then you have an antagonist. So this blocks the activity of a neurotransmitter system. And it can happen also as well at the level of receptor. So like naloxone comes and sits on an opioid receptor, so that way heroin can't come and bind there, right? So I guess my point is that there's many ways, levels at which there can be agonistic or antagonistic properties to something that either facilitates, right, the activity of a neurotransmitter system or antagonizes it. I always have students think about what is going to happen at the end. Like, is the receptor going to be activated and an ion channel open? If an ion channel is able to be open, no matter at what level there's intervention, it's an agonist. If it's not, then it's an antagonist. And so you can work through that in-class assignment there. And we'll be using those terms sort of throughout this lecture and a lot in next lecture when we talk about how exogenous chemicals affect our neurotransmitter systems. So let's go through the neurotransmitter systems. First, we're going to start with amino acid neurotransmitters. Then we're going to talk about neurotransmitters that derive from amino acids. And then we're going to talk about neurotransmitters that are strings of amino acids. So it's kind of all related. So GABA is our most widespread inhibitory neurotransmitter. It is ionotropic and it opens a chloride channel. So if it opens a chloride channel, think about that. Chloride's going to flow in. It's going to make the inside of the cell more negative. So it's going to lead us to an IPSP. And here you can see that there's a GABA site. There's sites for other things too, which we will talk about next lecture. But so it comes and it sits there and it opens the channel. So GABA is a agonist to this GABAergic cell or this GABAergic receptor because it does what GABA would normally do, which was leads us towards an iPSP. Even though it's inhibitory, that doesn't mean it's an antagonist because it doesn't antagonize, it doesn't block the action of this ion channel from opening. It facilitates it. It just happens to facilitate a chloride channel, which leads the inside to be more negative. So it's an agonist. I'm never going to say anything more about this, but there is another receptor, which is GABA, which is metabotropic, which opens a potassium channel. Potassium will flow out, and then the inside of the cell would become more negative, still leads to an iPSP. Glutamate is our most excitatory or most widespread excitatory neurotransmitter. So GABA is our most inhibitory, glutamate is our most excitatory neurotransmitter. And it has 3 receptors that we care about, NMDA, AMPA, and kainate, which are here. We're really only going to talk about NMDA and AMPA. So AMPA is ionotropic, and it lets in sodium. Now, NMDA is special. It has 6 different binding sites on it, one of which is glutamate, but it also has this block, his magnesium block. So unless the magnesium block can get kicked out, the channel can't open. Nothing can sort of get through it. So we talk about AMPA being chemically gated channel, which by that I just mean glutamate comes and binds and it opens. Not a chemically gated receptor. Where NMDA is both chemically and voltage gated. So there has to be a change in voltage in order for that magnesium block to pop out. And when it pops out, if and only if there's glutamate bound here, then will the ion channel open. And so how does that happen? It usually happens because it's sitting next to an AMPA receptor here. So the AMPA receptors will get activated by glutamate. The ion channel will open, sodium will come in. When it does that, it changes the voltage of the cell, just like it moves us closer to an EPSP. It changes that voltage of the cell. When it does that, it will pop out this magnesium block. And if glutamate's bound there, then it will open the ion channel and it will let in both sodium and calcium. Glycine, we're never really going to talk about it again, but it is also an amino acid neurotransmitter. It's inhibitory and it has ionotropic receptors. It's located in the central nervous system and primarily in the spine. So if anybody ever get like tetanus, they say you get lockjaw, it inhibits glycine. So if you inhibit the inhibition, then you have a lot of activation if you have less inhibition. Okay, let's talk about, take a break real quick and talk about acetylcholine. So acetylcholine is a quaternary amine, which means it has 4 amine groups on it. And it was one of the first neurotransmitter systems founded, likely because it's both in the peripheral nervous system and in the central nervous system with differing effects. So in the peripheral nervous system, it's really important for muscle activation. So in the PNS, at the neurotransmitter, NMJ stands for neuromuscular junction. I'll write that out for you. At the neuromuscular junction. And these receptors are called nicotinic receptors because nicotine activates them. Nicotine is a better agonist for them than actually acetylcholine is. And here you can see their how it goes on real muscle, which is pretty cool. So here's in the peripheral nervous system, here's our myelinated neuron here. So these would be Schwann cells, axon terminal. We see exocytosis has happened, the acetylcholine has been released. It's going to bind to these ionotropic receptors here, and they're going to open, and they're pretty fast opening. Acetylcholine is in most of the autonomic nervous system, which we talked about, at all of the preganglionic sites and at many of the postganglionic sites, except for here in the sympathetic nervous system, where this could be noradrenaline or nitric oxide. Think about operating that fight or flight system. Think about noradrenaline, which is also called norepinephrine. That's important for sort of stress-related factors, and so it's involved here with the sympathetic nervous system. Though here, it doesn't use nicotinic receptors. It uses what we call muscarinic receptors. And they're called that because the agonist for them is something called muscarine. And so for the most part in the peripheral nervous system, it is nicotinic receptors, except for here where there's muscarinic. In the central nervous system, it's mainly muscarinic receptors, except for the places where they're nicotinic, and the hippocampus happens to be one of them. So as I mentioned, acetylcholine in the central nervous system is mainly muscarinic and plays a role in learning, memory, and REM sleep. So a lot of Alzheimer's drugs try to target this. We see choline has been put into Flintstone vitamins for kids, for pregnant women, and you can facilitate, and REM sleep sort of facilitates learning. If you block an animal from having REM sleep and you teach them a maze and then block the REM sleep, they won't learn. So again, sort of activation of that cholinergic system seems to be really important for learning and memory and REM sleep, all which are tied together. Okay, so now we're going to go to the monoamines, which are derived directly from amino acids. So we have two kinds. We have catecholamines, which are going to be norepinephrine and dopamine, and then we're going to have serotonin, which is an indolamine. And they're labeled that way differently because they're derived from different amino acids. So let's start with dopamine, the first one. So dopamine comes from tyrosine. So I don't need you to know what the enzyme names are. I just need you to know it goes tyrosine to L-dopa to dopamine, right? So we have these enzymatic reactions that have to happen. You might have heard of L-dopa being treated for something, and that's usually Parkinson's, which we'll talk about when we talk about drugs. So dopamine, you sort of heard, is important for reward. It actually has three different systems that are important for three separate things. Reward happens to be one of them, but there's three of them. So the cell bodies for the dopamine lie in the dopaminergic nigrostriatal system, and it's called that because it goes from the substantia nigra, so that's the nigra part, to the striatum, the dorsal striatum, which happens to be the basal ganglia here. You remember the basal ganglia is important for movement and sort of motor memory, and that's what we have here. So things that are disorders of the basal ganglia are Parkinson's, movement issues, Huntington's, OCD, actually, so the repetitive movements and behaviors, and Tourette's, which are, sort of movement, tics, which involve movement and behavior. So as I said, Parkinson's is associated with dopamine neuronal degeneration, particularly in substantia nigra. And we know that the substantia nigra is named that because the breakdown products of dopamine are black, and so the substantia nigra is black. Except when they did post-mortem analysis of brains of individuals with Parkinson's, they found that it was white. So that means that they weren't having dopamine there. It can turn it black to break it down. So this is just a picture of Michael J. Fox, who's still alive and has Parkinson's. Muhammad Ali, who actually had traumatic brain injury induced Parkinson's from boxing. He passed away some years ago. Sorry about that. And so Parkinson's can be treated with L-dopa. And the reason they're treated with L-dopa as opposed to dopamine is L-dopa can cross the blood-brain barrier, can cross that astrocytic level sort of there. And so you can give L-dopa, and then there, once it's in the brain, it can be undergo an enzymatic reaction with dopa decarboxylase and then become dopamine. Okay, so that was the first system. That was our nigrostriatal system, which was movement. This one is a mesolimbic system, which I like. It's in a name as well. So this goes from the ventral tegmental area to the limbic system. So we're talking about things like the amygdala, the nucleus accumbens that are involved in reward and motivation. Also, drugs of abuse activate the system, releases dopamine 2 to 10 more times more than any sort of natural reward. And so it's a lot of things that help to keep us alive. Eating, having sex, orgasms, even holding hands, laughing, right? Keeping us sort of social, that sort of go towards the goal of life and biology, which is to pass on your genes. But just to show you, again, this goes from the ventral tegmental area. So here we're in the tegmentum. So ventral tegmental area, ventral, right? We're in the tegmentum, part of the midbrain. This area is very important for reward, and so is the nucleus accumbens here. So those are two main reward areas. Our next system is called the mesocortical system. So again, it's in the name. It goes from the ventral tegmental area to the neocortex, and that's sort of all of our things that make us cognitively unique. So planning, strategy, attention, all of that is going to affect our mesocortical system. If you prefer humans, so this was a rat brain. I guess I should orient you to that. So down here, you have the posterior piece. Here's the cerebellum. Here you have the olfactory bulb, and you're looking anterior. Here is a sagittal section of a human brain. So here's the brain stem right here, pons midbrain, where we see those dopaminergic cell bodies lying and going all over the brain. Now, norepinephrine is also a catecholamine, which means that it's actually derived from the same amino acid as dopamine is. So if you're British, they call it noradrenaline. I don't know why we have different names, but you will not hear the receptors referred to as norepinergic. They're referred to as noradrenergic. So that sort of comes from the noradrenaline. So norepinephrine is associated with various stress-related functions. Think about anything that is altered in stress: sleep, attention, vigilance, right? The norepinephrine is going to be important for those type of things. So you can see here that it actually is derived from dopamine. So if folks have a deficit in dopamine, you likely have a deficit in norepinephrine as well. Again, I don't necessarily need you to know that names of the enzymes, just they occur, but I do want you to know this: tyrosine, L-dopa, dopamine to norepinephrine. Skip that. The most important noradrenergic cell bodies are located in the locus ceruleus here. So remember, so this is in the pons, and it's very important for wakefulness and arousal. So think about that and when you're thinking about stress. And here you can see that it goes sort of all over the brain and actually goes back down to the spinal cord too. Again, if you like human brain, here you go, our sagittal section. Here's our locus ceruleus and our raphe here in the pons, and you see the projections going all over the brain and back to the spinal cord as well. So remember that acetylcholine was important for all the parts of the autonomic nervous system except for the postganglionic piece of the sympathetic nervous system, and that is norepinephrine. So right here you have that big norepinephrine. Again, think of that as sort of the fight and flight, and that norepinephrine is important for stress-related functions, and those things are related, and that can help you remember. So those are our catecholamines that were all derived from tyrosine. Now we have serotonin, which is an indolamine, which is derived from something else. And this is derived from tryptophan. So it becomes tryptophan, 5-hydroxytryptamine, and then 5-hydroxytryptamine. We call it serotonin. I don't actually know the history behind that. Or I've called 5-HT. So almost always I will abbreviate it as 5-HT. Most papers you see will abbreviate it as 5-HT. 5-HT and serotonin are the same thing. So serotonin is complex. Most of the neurotransmitters are. It's important for mood, eating, sleep, arousal, pain regulation, actually dreaming too. And so things like ecstasy and LSD are serotonergic agonists, which mean, and I guess some antagonists too, but that accounts for the visual effects that folks see when they take sort of those medications. So here again, you're looking at, we're in the brainstem. Here's your RAF nuclei where most of your serotonin clusters are. They're going to go all over the brain and also back towards the spinal cord. Again, if you're into humans, you can see that there's lots of cell bodies that are going to line the brainstem here. Okay, so now we talked about amino acids. We talked about catecholamine or monoamines, sorry. So that means they're derived from one amino acid. Now we're going to talk about peptides, which are strings of amino acids. So these are actually made in the cell body, and they're transported down the axon via axoplasmic transport, excuse me. So in the cell body, you'll have this huge, long, it almost looks like a string of pearls. And in it are multiple, like if you cleave it apart, you'll get multiple peptides from them. So depending on how many there are and what order they are, there'll be different peptides, even if they're related. So for example, I did my dissertation on corticotropin releasing hormone, which we'll talk about when we talk a little bit about the stress and homeostasis. I think that is a 38 chain of amino acids. I also did experiments with urocortin, which is related to it, which is in the same exact order, but it has 41 amino acids. So it just has like a 3 difference. So again, depending on how many and what order they're in and where this gets cleaved, you're going to get a different peptide that's then going to get shot down to down the axon to the axon terminal. Okay. So our most common peptide system, there's lots of them, so don't get me wrong, there's lots of them. This just happens to be one of them, and our most, I don't know if it's our most common, but it's our most well-known, are opioids. So they're involved in analgesia and reward. So that means sort of pain relief, right, and rewarding. So things like enkephalins, endorphins. So I don't know where you've heard endorphins for, like running, right, that you feel good afterwards. Why would anybody do it if you didn't feel good afterwards? and it sort of has pain relief associated. Otherwise, it would be really painful, right? Then we have exogenous opiates, which are like opium, morphine, heroin, codeine, all those types of things, which are agonists, so they're going to act on the system. Whereas naloxone will sit and block that system. My battery's running low. For an agonist. This is just showing you a picture of a coronal section of a rat brain, and it's highlighting for you all of the opioid receptors. And so that is in the ventral striatum, which is the limbic system. So we know that the limbic system is important for reward. So you can see how many areas can get activated there in response to the opioid system being activated. We have other neurotransmitter systems called gaseous neurotransmitters. So nitric oxide happens to be one of those. And that means it is made on demand and it can pass through cell walls and it doesn't last for very long. So nitric oxide is an example of this, and it dilates blood vessels. So for example, nitric oxide or Viagra activates nitric oxide. So what Viagra does, it helps with erectile dysfunction. It doesn't have any effect on libido, but it helps with erectile dysfunction. All erections is blood getting to the penis or blood getting to the clitoris, right? And so when that Viagra allows for that to happen, and then a person could get an erection. And it only lasts for so long. They only take it 4 hours or whatever, because again, it's the gaseous neurotransmitter, and it lasts. Okay, that is the end of that lecture for today. Make sure that you answer the neurotransmitter questions online.